the solution here was to completely throw out everything that we knew about physical chemistry and instead replace it with deep learning.
Hey, everyone. Thanks for joining us. Today's conversation, we're going to be in the realm of biology, computational biology, all the wonderful work that's being done to synthesize new proteins for various purposes. And I'm pleased that we have one of the primary architects, primary leaders of that whole scientific endeavor, who is David Baker. He's a biochemist, a computational biologist who heads the Institute for Protein Design at the University of Washington. He shared the 2024 Nobel Prize in Chemistry, and he's joining us now from his laboratory in Seattle. So welcome, David. Thanks so much for joining us.
Oh, thanks, Brian. It's great to be here.
Now, that is a real laboratory behind you, right? This is not some green screen.
It is a real laboratory, and you'll hear some background noise from instruments and
people
talking time to time.
Yeah, maybe love to hear that. And were we the same class in college?
Yep.
Is that correct? Yeah. Yep. And so what house were you in?
I was in Quincy.
Back at Harvard. You're at Quincy House. Okay. And if I'm not mistaken, your major was not necessarily biology, right?
Yeah, I became a biology major in my last year. I was actually a social studies major and then briefly a philosophy major before seeing the light and switching to biology in my last year.
It's funny because that's the unusual direction to go, right? Many kids come into college thinking they want to be a physicist, a chemist, biologist, whatever, and then the going gets a little bit rough as these subjects are pretty intense, and then they switch out to maybe philosophy or so. It's not that those are easy subjects, but that's kind of a familiar pattern. How did yours go the reverse?
Well, I think what attracted me to biology, I took a developmental biology course in my last year in college, and that's really what inspired me. And just the idea that there was a clear forward direction, that things were moving. I mean, with social studies and philosophy, things were pretty static and not much had changed. And, things were kind of devolving into language games, which was a little frustrating. So the idea of an activity where, progress was being made, that you could jump in and push the frontier was really attractive. And when I actually started doing it in graduate school and later, it turned out to be really fun. And I've loved it. We're very much engaged in that today. So
Certainly
new discovery, exploring frontiers, that kind of thing.
Right. I mean, it's worked out well, obviously. But just for even clarity, did you have a lot of catching up to do when you decided to make that move, or you were always in the background filling in?
I took probably every intro course at Harvard. I really didn't know what I wanted to do. I think you even helped with my math homework at one point.
Someone mentioned that to me. Yeah. I hope I got the answers correct.
I'm sure you did.
Which is really good to hear, of course, because my son is at Columbia, and he describes how the student body today is so focused on knowing exactly what you want to do from the get-go. And most of them want to do finance from the get-go. And it just sort of, in a way, breaks my heart that people are not exploring the realm of sort of human endeavor, the way that you can in college, and just see what really speaks to you. But clearly, I guess that was the route that you followed.
Yeah, and I'm a complete believer in that. I think people specialize too early. Even really extensive undergraduate research, I think, can be a little bit of a distraction from getting, a broad view of things. And I think it's, when I first started doing research in graduate school, I thought that all that other stuff I'd done in college wasn't really relevant. But, as I got deeper into things, I think it actually was very useful. I mean, there's a lot of science, as can also devolve into language games, and being able to recognize substance from talk is really an important thing. And obviously there's a lot of writing and communicating that's involved as well.
Yeah. And so when you started in graduate school, was that immediately after graduating from Harvard, or did you have a break or anything?
I took a year off. I biked across the country, spent some time working and kind of traveling around Asia, and then I started graduate school the next year.
Right. And so was there a primary exciting area in?
Yeah, well, I thought when I started graduate school, I thought that I was interested in developmental biology and neurobiology, how the brain functions. But when I got to graduate school, I found out that things move sort of slow in those areas, and you had to cut up animals, which didn't sound like fun to me. And so I got interested in how organization happens within cells. And so that's basically what I studied for my PhD.
I gather if we just jump into some of the details, I mean, my knowledge of biology is certainly at a very rudimentary level, but I gather. I mean, the way I think about it, and do correct me if I'm wrong, is that basically proteins are these little miniature workhorses of the cell that sort of get the jobs done that a cell needs in order to survive and thrive. And a lot of work has been done over the course of decades now, understanding how, genetic sequences of the 4 nucleotides in DNA get transmitted by RNA to the ribosomes that then synthesize these proteins, which are then distributed throughout the cell doing what needs to be done. Is that sort of the basic architecture within which you are working?
Yeah, that's exactly right. So just to clarify, as a graduate student, I didn't work with proteins at all. I was working with cells. But then towards the end of that, I became interested in really the sort of fundamental question of self-organization in biology. I'd been looking at it at the level of cells, and I got very interested in looking at the level of individual proteins, like how do proteins fold up? And what you described in terms of what proteins do is exactly correct. So proteins are essentially the machines that do all the important things in biology, and the genome, our genomes and the genomes of all living things simply provide the instructions, the blueprints for making them.
Again, one of the striking factoids, I guess, is that the difference in the genomes of individuals within the same species differ by a very tiny amount. And so for the most part, we're pretty much the same. And even if you go to nearby species, you're talking about what, a 1% or 2% difference between us and the chimps, or you can correct my numbers. But basically, the genetic information is very similar across species that are at sort of the same point in the developmental tree of biology.
Yeah, that's right. I mean, very subtle differences in sequence are responsible for at least parts of the differences between us and chimpanzees and other animals. So one of the things that is interesting about proteins that anyone who's taken a biology class knows is that basically all the proteins we've known, like if you take a biology class, you learn about proteins and they have these exotic names like hunchback or crippled or Cas9 or whatever. And each one has been lovingly studied by a scientist, often for their entire career. And biology just is a very descriptive subject. So you have all these exotic names, and proteins sort of came about through evolution, millions or billions of years of natural selection. So they're sort of like elven runes, that sort of like in the Lord of the Rings, you have these magic things from the past that are handed down that still have these magical properties today, like some special type of sword or something. And I think that's really how biologists and biochemists have looked at proteins for a very long time. They're these magical things. They've been passed down from the deep, dark past, and now they're here and they do these amazing things, and we can study them and figure out how they work. What I was sort of fascinated by was the idea of could we actually make brand new ones to do new things? But I think to appreciate that, you sort of have to understand that in biology, it's basically the study of all these marvelous things that came through evolution.
And so just to get a sense of scale, which I don't really have in mind, how many distinct proteins have been lovingly studied by scientists over the course of generations?
That's a good question. So we now know how many proteins there are in a typical genome, like the human genome. And if we leave out subtle modifications, they're on the order of 20 to 30,000 proteins, distinct proteins in humans. Now, every organism, will have its own complement of proteins. As you said, like monkeys will have more similar proteins to us. Jellyfish will have very different ones. And not every one of those proteins has been carefully studied. So many of them have names, which means that someone studied them and named them. Others just have numbers associated with them when people don't know quite what they do. So we now sort of have an idea of the complement of proteins in humans and in all the animals and organisms and plants whose genomes have been sequenced.
So it's on the orders of tens of thousands. Is that a reasonable way of thinking about it?
Yeah. I mean, across all the organisms now there's, as you know, these high throughput genome sequencing has become very widely used. And so we now know the genome sequences of thousands, probably tens of thousands or more different organisms. And each one may have 10 to 100,000 proteins. So the total number of proteins whose sequence we know is very, very large. the number where a scientist has really studied them and knows what they do is much smaller. There's a lot of argument by analogy. So as you said, the proteins in one species can often be closely related to those in another. So if a scientist has studied and figured out exactly what a protein does in one organism, and you find a related one in another, then you can sort of transfer the functional information.
And do you find that that is the case? Is functionality fairly consistent for a given protein? If you find that protein in different species, it typically does the same thing in each of them?
Yeah, one of the really remarkable observations that was made about the time I was in graduate school, I wasn't involved at all, was that the genes involved in sort of specifying the body plan in insects were the same as those in vertebrates, which was really quite astounding. So there is a lot. So proteins, even in very different organisms, can have the same sort of similar overall roles.
And so is there a sort of typical number of amino acids that make up these proteins, or does that have a wide range?
There's a wide range. It's typically from about 50 amino acids to on the order of 1,000 amino acids. So the longest proteins, or one of the longest proteins, is kind of interesting. It's called titin, and it's in muscle cells. And muscles have to have a lot of tensile strength. So it's very, very long protein that goes from cell to cell and gives muscle its sort of ability to stretch to some extent. So yeah, between 50 and 1,000 amino acids.
And I gather too that the functionality of a given protein is highly dependent on the 3D shape into which the sequence of amino acids folds. And so to what extent is that the dominant way of determining the function of a protein versus understanding the real detailed molecular makeup? I mean, I tend to go to the fundamental ingredients, right? I'd love to go down to the level of quarks and electrons when I think about any molecular system. To what extent do those details matter, or do they get superseded by the 3D shape of the particular protein itself?
Well, the fundamental constituents of proteins, at some level, it's the quarks and so forth that constitute the atoms. At another level, it's the atoms. At a higher level, it's the amino acids. Those are all the same between these. I said 10,000 genomes, 100,000, 10,000 proteins each. So we're talking about billions of proteins that are known. The fundamental building blocks are all the same. The amino acids are all the same. The only difference is the order in the sequence. So each protein has, is a linear sequence of amino acids. So the things that uniquely define a protein and differentiate it from other proteins is the amino acid sequence. And as you said, that amino acid sequence generally specifies a unique three-dimensional structure.
And understanding that three-dimensional structure has been a long-standing issue that only in recent times, I gather, your fellow Nobel laureates have helped elucidate understanding, given the sequence of amino acids, what will be the 3D shape into which that folds. So can you describe the difficulty in cracking that problem, and also the importance of cracking that problem?
Right. So starting in the 60s, scientists have worked to solve the three-dimensional structures of proteins. And so over the years, they're now on the order of 200,000 structures that have been solved of different proteins from different species. And that's done in several different ways. One way is to grow crystals. So you take your protein, put it in solution, a crystal forms, and then you shoot X-rays at the crystals, and you collect the diffraction pattern, and you can back out what the three-dimensional structure is. More recently, cryo-electron microscopy has become very powerful for larger proteins, and you can ultimately assign where all the amino acids are in a structure. Those are very they're quite laborious techniques. It used to be that a graduate student might spend their graduate career determining the structure of one protein. So fundamentally, actually, I think we have an animation of this. So
perfect.
So what you see on the left is a protein chain, and the little spindly things coming off the chain are the side chains. Those are the different amino acids. And so when I first started my job here at the University of Washington, one of the problems we worked on, we were really interested in how this folding process took place. So we were did a series of experiments to try and work it out, and then we incorporated the insights into actually structure simulation methods like you see here. So the idea is that from a sort of physics point of view, proteins fold to their lowest energy state, and so the challenge of predicting the structure from amino acid sequence is given the sequence of amino acids, find the lowest energy state. And that sort of a schematic of that is shown on the right. There's obviously a huge loss in entropy when a protein folds, and that's made up, compensated by the formation of all the interactions between the atoms in the final folded state. And so we made some progress on that problem, and for simple proteins could predict the structure from the sequence. And we could tell whether we were right, because as I said, there's a very large number of proteins whose structures have been solved. So we could take their amino acid sequences and determine whether this kind of folding simulation search for the lowest energy state could predict the right structure. But the methods, our methods, and then other people developed methods as well, were never all that great. And the reason it was difficult is proteins are really large molecules. They're thousands of atoms. A very large protein can have tens of thousands of atoms. And so you have the global optimization problem. So you got to find the lowest energy state of a system with, say 20,000 atoms is really, really hard. So there's the computational sampling problem. There's also the problem of accurately calculating energies. And so in this sort of system, it's too large for any kind of quantum chemistry calculation. So we and others developed sort of empirical force fields where you're modeling the various interactions that take place, the electrostatic interactions, the van der Waals interactions. Hydrogen bonding turns out to be very important. And so we were able to make sort of improvements to both the conformational sampling methods, and computers got more powerful so we could do that better. And we were able to model the interactions within proteins more and more precisely. But still, even by the end of, 2018, we still could not reliably predict the structure of proteins from their sequence. So this is where DeepMind and my fellow laureates, John Jumper and Demis Hassivas, entered in. So they took a completely different approach. They threw physics out the window and simply took advantage of the fact that scientists had solved the structures of hundreds of thousands of proteins. And so basically they developed a network, AlphaFold, that takes takes as input the amino acid sequence of a protein and then outputs the three-dimensional structure. And you can think of it as just fitting a function. So it's a mapping from sequence to structure. You've got a couple 100,000 examples. And so you fit the parameters of the model to so that on a training set of structures, you can robustly go from sequence to structure. And then the question is, what happens does it generalize? Can you give it new sequences and have it accurately predict the structure of those sequences? And I think it wasn't so surprising that you could have a model learn to predict the structures of naturally occurring proteins. In fact, scientists had done that many, many times. What was remarkable about AlphaFold is that it generalized. And it's really quite interesting, particularly for people with an interest in physics, because the solution here was to completely throw out everything that we knew about physical chemistry and instead replace it with deep learning.
Yeah, that's the remarkable thing when I read about this. I mean, I think many of our audience are certainly familiar with this at the level of, say, large language models, where you can say, we're throwing out everything to do with grammar and syntax and all that good stuff. If you have enough data, the patterns emerge and the system can create beautiful prose. Well, prose, let me not call it beautiful. There's an argument there, but it can create, acceptable prose even without understanding the architecture of language. And here you're saying, even without understanding the architecture of the basic physics by which, one atom pushes against or pulls against another, you can still get the structure into which this complex sequence will fold, which, I mean, did you think that program was going to be as successful as it proved to be when you were hearing about it?
Let's see. Well, if I had thought that deep learning was going to solve the problem, we would have been focused on that. And
sure.
So I was, I was as surprised as everybody else. I thought it was super exciting that. The thing that's not obvious is, if you have enough data, you could imagine if with given enough free parameters, you could learn anything. The question is, how much is enough? And
Yeah.
my intuition had been that there was not enough data yet, but there clearly was. And if you set up models more cleverly, they have more inductive bias. So you need less training data to accurately sort of describe the space that you're trying to, the mapping. But I found that really, really exciting. So when DeepMind presented their results, we immediately jumped into deep learning. We had to learn what that was and become proficient at it. And we had been working on protein design. And so when we saw the amazing results DeepMind was getting with protein structure prediction, we immediately set out to develop deep learning methods for designing proteins.
And so before you went in that direction, what was the day-to-day work of trying to design a new protein like? Presumably you had not thrown physics out the window.
No.
Yeah.
In fact, if you remember the animation I showed with where we are trying to predict the structure by finding the lowest energy state for a fixed sequence, the protein design problem we approached in exactly the opposite way. So starting back more than 20 years ago now, after basically what happened was after we had some success in predicting structure from amino acid sequence, we realized we could go backwards and take a new structure, or build a model of a new structure on the computer and work backwards towards the amino acid sequence which encoded it. And the principle was exactly the same. We had to search for an amino acid sequence whose lowest energy state was the structure we wanted to make. And
on the face of it, it seems like an over-constrained problem naively when I hear about that. Is it not like if you wrote down any structure, how do you know there is an amino acid sequence for which there is a lowest energy state that gives you that structure?
Yeah, that's a really good question. So the first problem is, how do you generate a structure which is designable in the sense that you just said, that there exists a sequence that could fold to it? So the first problem was, how do you generate plausible structures? Well, as I said, we've been working on protein structure prediction, so we already had methods for sampling through plausible structures. We simply had to extend them to the case where you don't actually know the amino acid sequence, so that the structures you generate are plausible. That was just that was sort of the problem of generating new proteins that didn't exist in nature. And this was relevant because, coming back to that Elvin Rune example I gave earlier, now the demonstration that you could actually make new proteins that you could make in the lab and that folded up into new structures was an important milestone, because it then opened the door to designing proteins with new functions. I actually have schematics. So in biology, this is sort of comparing the flow, how information flows in biology. So we have genes in our genomes, that's at the top left. These encode amino acid sequences, which then fold up into proteins, which have biological functions. And then in the design case, we run this in reverse. So we start off with a new function that doesn't exist, and we'll get to that a little bit later. So you have a new function, then you reason about, well, what shape protein would be likely to have this function. So you make a protein with that structure. It doesn't exist. This is on the computer. You work backwards to the amino acid sequence. So now we're on the right side. And then once you have the amino acid sequence, you work back to the sequence of DNA that would encode it. But now there's an issue, because this is a brand new protein, so it's a brand new gene. We can't find that gene in any extant organism. So instead, we make a piece of synthetic DNA that has that sequence. Once we have that piece of synthetic DNA, we put it into bacteria. The bacteria become factories to make the protein, and we can then extract it and determine whether it folds up to the structure we were aiming for, and whether it has the function that we wanted.
Just one point of clarification there. So when you have the bacteria become this synthesizer of this new amino acid sequence, hopefully that folds up into the protein whose desired function set this journey off in the first place, how many other proteins does the bacteria produce? And how do you pull out only the proteins that are of interest to you from this synthetic approach?
Yeah. So we can make the bacteria into essentially a factory for making the protein of interest. we can rig it so the major protein product of the bacteria is the protein we want. And we can put a tag on it so that it's easy to separate that protein from all the other stuff in the bacteria after we break them open.
Got it. And just quick, I may it's probably too technical. What does that tag look like? How does that work?
There are a variety of tags that can be used. One common tag is a short amino acid sequence that binds to metal ions, and so you basically have an affinity matrix with a metal ion on it, and that the tag binds to that.
And then you pull it out using
you
that
just
as
literally pull it out, right? Yeah.
Got it. Okay. And so this is all on a interesting computational abstract level. Obviously, you've put this into practice. Do you have any examples where you've successfully yielded the desired proteins?
Yeah. So we've probably designed we've designed millions of proteins at this
Millions?
stage. And
really?
Yes.
Really,
So
millions? David,
really
that's not
millions.
exaggerate. Yeah. Wow.
No. So, in fact, the reason we initially designed large numbers is our design methods were not very like our prediction methods were not terribly accurate. So, one of the first things we tried to do, for example, is design proteins that would block influenza virus from infecting cells. And so we developed methods for designing not just one protein, but 10,000 candidate proteins or 100,000 candidate proteins, and very rapidly testing them to see which ones actually blocked influenza virus. So a more recent example is shown here. So the proteins in gray are components of snake venom, and the colored proteins above are our designs. And here we made far smaller numbers. This is work from a super talented graduate student, Susana Vasquez-Torres, who made on the order of 90 designs for each of these and was able to find binders that were quite tight in both cases.
Wow.
And so these are really neat proteins. If animals are animals who are exposed to the toxin, die very quickly, whereas if they're given these proteins shortly after they're exposed to the toxin, they live. So we're very excited about these as new sort of antivenoms.
And are these used in the world? Are these
so we're trying. There's always this step between designing new molecules like these and getting them out in the world. And so so Susanna is working on that now. We have started a number of companies that are taking design proteins and developing them. There isn't a huge commercial market for better antivenoms. It's usually in developing countries where snakebite is a problem, and so there's some sociology that determines whether or not what sorts of proteins it's easier to actually translate. And
do you have any connection? This is probably off topic, but do you have any connection to? I've read the story about some guy who's getting bit by basically every snake in the world to try to get the universal snake venom antidote. Is that at all
well, that
relevant?
would be a different approach. So that would be that's probably more a conventional approach where you take antibodies. So the way that antivenoms are made now is you inject the venom into a horse, and the horse makes antibodies against it, and then you get horse serum. But it has to be kept cold. It's complicated. It can be a little dangerous. So the idea is that these little tiny proteins we design are they're much more stable. They're much easier to make. You don't need a cold chain. So that's sort of this would be sort of a next generation, probably better approach. So we've made binding proteins like this to probably over 300 different protein targets now. So that problem of how you design proteins to bind to other proteins, I consider pretty much solved.
And can you just flesh that out a little bit, just so I have a mental image in mind? Is this sort of a 3D structure modeling problem? And do you have a target that has a certain shape and you're basically just trying to grab it with a complementary structure that can lock to it?
Right. So we left out one step though, which was, as I said, when we started off designing proteins, we were using physically based methods. If we wanted to make a protein which bound to a target like the influenza virus, we basically we came up with a shape that was complementary to the viral protein, and then we designed a sequence we predicted to fold up to that shape. But as I said, when DeepMind presented AlphaFold, we immediately went into high gear with deep learning methods. As DeepMind showed for structure prediction, we found shortly afterwards, held for protein design as well, that the deep learning methods were far more powerful than the previous physically based methods. And so what you see, and the method we used is are broadly generative AI methods. They go by names of diffusion or flow matching. And so what you see here is a diffusion trajectory for binder to the insulin receptor. And it's just what you said. The insulin receptor is in gray, and it has a particular shape. And the method starts from a completely random distribution of amino acids, and then progressive denoising steps lead to the final structure, which is just like you said, is complementary in shape to the target.
And presumably there are many solutions because the target has such a corrugated structure itself that you could bind in many different places in many different ways. Is that correct?
That's absolutely correct. And so there are many different solutions, and that's why we could make and test many different designs against the same target.
Perhaps this is more of a philosophical question, but do you feel you understand the synthetic protein as well as you do when it emerges from these generative AI methodologies, deep learning methods, versus when you sort of understood sort of the physics of how the pieces were pulling and pushing and created that structure?
It's a good question. And this is something, the interpretability of deep learning models is obviously a big question today. I would say that first of all, the design proteins, whether they're generated using physically based methods or deep learning methods, are generally much simpler and easier to understand than naturally occurring proteins, which tend to be much larger and more complicated, and have a lot of features that are probably evolutionary accidents. Whereas the design proteins are they're minimalist solutions to the problem. I think in some sense, if you are a physically based model, does give more interpretable results. But on the other hand, with the deep learning models giving more accurate, robust designs, that trade-off is definitely worth it. And people complain about neural networks, but we're all accustomed to using very powerful neural networks every day. Like currently, you and I are having this conversation. We're not worrying about exactly how our brains are processing the information or giving rise to speech. So, it's something that every human is deeply accustomed to with, and we don't get into existential crises about not understanding exactly about how our brains work.
Although some of us do at times, I'll say.
Yes,
but yes.
Some of us do at times. The audience might be interested in how this is actually done. This is how we train generative AI models for protein design, and it's inspired what people have done, how image generation works with AI with methods like DALL-E. So you start with a large collection of images on the internet. This is in the DALL-E case.
Yep.
And you noise them to different extents, and that's shown on the top. And then you train a network to, given a noised image, predict the ground truth unnoised image. And all you give it is the noised image and the amount of, you tell the model how much noise was added.
Yep.
And once you've done this, it's quite remarkable how well this works. You can start with with random noise, and then progressively denoise it, hand it to the model, specifying what the amount of noise is. And you can sort of see intuitively that if you stay in distribution, like at each stage of denoising, you really generate an image that looks like it came from the slightly less noised training set. Then what you end up with after complete denoising is an image. It looks like a member of the training set, but in fact, it's completely new. And so we did exactly the same thing with proteins. I told you about the large set of protein structures whose structures are known, proteins whose structures are known. So we take every one of those structures, we noise it to different extents, we train a model to denoise, to basically take the noise structure and predict what the ground truth structure was. And then at the end, after we've done that, we can start with complete random noise, and that's shown on the bottom left here, and progressively denoise, and we end up with a structure that looks like it could be a real protein structure, but actually it doesn't look. It's not anything that was previously seen before, at least when you look in detail. And so that's just how you generate structures. But just as in the case of images, generating new images isn't very interesting. You want to condition it. And so obviously in the image case, you can say generate an image of a cat sitting on a table, and that's what you get. And you do that by training the model with where you give it text. And so it learns to associate text with the final output. And we do the exact same thing here, where you can condition the model in many different ways. For example, in that insulin receptor diffusion animation, the model was conditioned on the presence of the insulin receptor target. So the model has been trained where it's given some part of a structure kept fixed, and so it knows how to generate molecules that have high probability to bind the target you're interested in. So another thing that proteins do is they make and break chemical bonds. And there's a lot of important uses for this now, ranging from fixing greenhouse gases like methane to breaking down molecules like plastic. And so here, what we do is we start with a description of a catalytic site, something that can carry out chemistry, and we build up a protein around it. And that's what you're seeing in the animation. The things shown at the beginning, that's the sort of ideal active site we're trying to make that's going to do the chemistry, and then we build a protein around it. And we've been able to make proteins that make and break quite a variety of different chemical reactions, which is kind of expanding the scope of problems we can now tackle with protein design.
I see. And are there specific problems that you've applied this to?
Yes, there are quite a number of problems. Thus far, what we've been successful with are primarily kind of model reactions where breaking down molecules in what are called hydrolysis reactions. We also can make chemical bonds. What we're applying these methods to now, I sort of already alluded to breaking down plastics and other polymers, to fixing methane, to coming up with greener routes to complex molecules, because we can design. So for drugs like pharmaceuticals are often big, complicated molecules. So we're now designing enzymes that can lower the cost of making such molecules. So there's really wide range of application. Another problem which we've recently succeeded with is designing proteins that actually chop other proteins into pieces. And this is very exciting from the point of view of therapeutics. So a lot of the way that modern medicines work, protein medicines like antibodies, is you get them injected and then they block a particular interaction. Like they block the suppression of immune responses that tumor cells use to evade the immune system. But you need large amounts of these drugs, and that's why part of why they're so expensive and why taking them is such, it can be really intrusive. So we're working on a next class of medicines where they not only recognize a target, but then they modify it in some way, for example, by degrading it. And there you should be able to we expect to get by with much lower amounts of drug because they act catalytically.
And so are you working with like medical schools and research folks who are directly looking for clinical applications? I mean, are you working on cancer, or is that?
So in fact, it's fun. In my research group, I have medical students, MD, PhD students who are really interested in medical applications. I have physicists who are interested in simply building better models, and computer scientists. There are people here working on trying to make plants more thermotolerant. Deal with
I mean, for global warming stuff.
Yeah.
Okay.
Yeah. So, and then in any specific area, we also pull in collaborators from around the world. Sort of my operating vision for the lab is what I call communal brain, with everyone, with a pretty large research group. Everyone's talking to everybody, and there are all these different problems that we can tackle, and then we pull in experts from around the world. So we've actually had proteins, cancer therapeutics in clinical trials. My colleague Neil King here at the Institute for Protein Design has develop vaccines that are actually in use in humans. And we're working on autoimmunity. I already mentioned infectious disease. And now sustainability is becoming an increasing interest, like with the catalysts I described.
So how big of a group is this?
So the institute is about 250 people. There are 5 faculty. My group is quite large. It's about 100 graduate students and postdocs, and it's just the range of problems that we can solve now with protein design is super exciting. So some people in the group are pushing forward to develop more and more powerful design methods, and then others are applying these to this really wide range of problems in medicine, technology, and sustainability. And it's just super exciting. And every week a new problem will come onto our radar screen, and yeah, there's a lot of really neat things to be done now.
So you made reference to the fact that the proteins in a living system emerge through a long period of evolution by natural selection, which, notwithstanding the fact that some people talk about an intelligent designer is not necessarily the most intelligent design, right? It's just some happenstance that yielded particular proteins that are able to do the job. But like you say, they can have portions of them that are an evolutionary baggage that you don't really need, and you can do things more efficiently. Have you experimented with taking simple life forms and trying to make them more efficient? I mean, taking out the proteins that nature has provided and replacing them by the proteins that you can synthesize that can do the same job, but without the baggage?
Yeah, so doing that's a really interesting idea. We've talked a lot about that. Doing that on systems-wide level is challenging because you need to replace out all the different proteins in a cell at the same time. If you just replace them one by one, then you have the challenge of, are they interoperable? Do they interact with each other in the right ways? But the thermal tolerization of plants is exactly that idea, that to replace the proteins in plants that are responsible for, that are not functioning well at higher temperature, and replace them with better versions. So before de novo protein design was developed, and even to the present, if people wanted to solve a problem in biology, or really wanted to solve a problem with proteins, there's the basic strategy was do bioprospecting, look in nature for the protein that does the most similar thing, and then do laboratory evolution to try and modify it to have the desired function. The problem with that is all the evolutionary baggage that you mentioned. You get all this other stuff coming along for the ride. So the neat thing about de novo design is you can design a new protein that does exactly what you want it to do and doesn't do anything else. And so you get more economical, more precise solutions. And so we're sort of seeing a transition in technology now where, I sometimes liken it to leaving the Stone Age. So during the Stone Age, humans took things that they found outside their caves, like, sticks and stones. They modified them a little bit to solve the problem at hand. And in human technology now, we really don't do that, outside the biological realm at all. If you want to build a building, you don't look around for for trees with the right shape. You build it from first principles. And so now with protein design, we can now start making proteins in that, build proteins up from the ground up that have exactly the properties and functions we want.
And so where do you stand on those plants that are better able to persist in higher temperature
Oh, we're just
settings?
getting going on that. Yeah. So that's, yeah.
But the vectors are pointing in the right direction. You see
What we've certainly found is that we can take proteins individually and make them far more stable and make them function better. This is sort of like, you asked about doing this at sort of the whole organism level. We haven't done that, but we've done that at the individual protein level. So we have very robust ways of taking individual proteins and stabilizing them. The challenge with plants is you have to know exactly which ones are the ones that you need to stabilize.
Right. And so when you do think more system-wide, one of course, the things that we see in science fiction or in fantastic literature is the construction of living systems, from scratch. I presume that you have the perspective that many scientists do, that in the end of the day, a living system is just a highly organized collection of molecular components. There's nothing else beyond necessary for a living system to be alive. And given that perspective, where do you think we stand on being able to one day start with inanimate raw materials and construct something that we would describe as being alive?
Yeah, I think we're not far off. I mean, to making simple systems, there are scientists now who are making quite a bit of advances on constructing completely synthetic systems that can replicate and carry out, simple molecular transactions. I think making things that are as complex as even a bacterium is quite a challenge, just because of all the different parts you need to worry about at the same time. But making self-replicating systems, there's considerable progress being made in that area.
Which, of course, is the first key step. How often have you encountered, and I guess this must be part of the clinical trial stage, but perhaps you've encountered it even before that, where you create some new protein and it does the function that you anticipated, which is why you designed it that way in the first place. But then there's some unintended consequence, some other thing that it does that is not what you wanted to also have happen.
Right. So that is, as you said, part of the part of the sort of testing that comes before a clinical trial. I described sort of the basic workflow earlier. So we have some function we want to make. We reason about what sort of shape protein would have that function. We make the protein, we make a gene, we produce it in bacteria, and then we have the protein. So we then test to see whether it has the desired function. And, whereas initially the fraction of the time that we had the desired function was small, now it's getting higher and higher every year or every week almost. So as far as testing what else the protein does and possible off-target things, we have a really neat collaboration with a research group in Europe, which can actually look at you can give them a protein. They can tell you exactly what proteins in a cell or tissue it interacts with. And so we're doing that now with hundreds of designed proteins to see what else they interact with. And most of them only interact with the desired target protein. There are some that interact with other proteins. And so that's telling us that, we need some more refinement still to get even higher specificity. In the clinical trials that have been done, and in the many experiments that have been done looking at sort of cancer and autoimmune and infectious disease models that we've done with designed proteins, we've, I think, almost never seen unanticipated off-target toxicity. The problem that we've seen is, and this happened with our cancer therapeutics in clinical trials, is that the designed proteins can cause problems, but it's not because they're interacting with something unanticipated. It's because, so in a cancer therapeutic, in immunotherapy, the idea is you stimulate the immune system to attack cancer. And the problem with that is you can have a byproduct that you also just get systemic, toxicity, because you're amping up the immune system. So we've seen kind of on-target toxicity, as has been seen with conventional immunotherapies. So we're now working on a next generation of immunotherapies where the immune system is only activated right in the vicinity of the tumor. So incorporating logic gates into these design systems.
Right. And so that collaboration with the group in Europe, are they working with a computational system that predicts whether your protein will interact with any other proteins in
No, this is experimental.
They're actually doing it.
Yeah. No, I mean, predictions are only, you need to do experiments to answer this sort of question.
So they actually take your synthesized protein and they inject it in a biological system and are able to track where that new protein is binding.
So they take the designed protein, they mix it with all human proteins, and then they use mass spectrometry to determine what's sticking to the design protein.
Well, yeah, got it. Amazing. And so you started out noting that initially when you went to graduate school, you were thinking of maybe more neuroscience and the mind, and there wasn't sort of all that much happening in that field. Do you still feel that way about that arena? I mean, do you see yourself migrating at all back to perhaps what initially drew you to the biological sciences?
Well, I think it is, how the brain functions is incredibly fascinating. And we have gone back in that direction. We're doing quite a bit of work now on neurodegenerative disease, trying to basically block, the amyloid fibrils from forming, and they're major players like tau and A beta, and we're developing methods. And one of the things I'm really excited about now is designing machines. So in biology, we have proteins that will take a chemical fuel, and that chemical fuel will drive a cycle between states, which then does work on the outside environment. And inside our cells, we have proteins called chaperones, which are basically like quality control things. They fix broken stuff by using a fuel to cycle between states. And whereas we have elaborate machineries inside cells, outside cells, we don't have such things because there's no, the fuel, which is ATP in biology, doesn't really exist outside cells. So what we're working on now are a next generation of machines, which will be driven by a fuel that can be in your diet, that then drives the machine through different states. So it can do things like, dissolve amyloid plaques or, unclog arteries. But coming back to your question, so we're really interested in applying these kinds of machines and other designs to neurodegenerative disease. And there's also some interesting ways in which, you can sort of interact with the brain. There's a lot of, interest now in brain-computer interfaces, where design proteins could play an exciting role too. So I think, we are coming back to the brain now after many years.
And so do you think brain-machine interface, I mean, I've seen rudimentary examples. I think we've all sort of seen the YouTube videos, which are incredibly impressive, where people are able to do various things through the process of thought. Where do you see specifically designed proteins aiding in that? How do you see that going?
In general, the interface between electronics and standard human technology and biology has been hard. So designed proteins, are things that can, you could imagine having designed proteins that both can interact with signals from standard, machinery, things that we would build, and a biological system. So I think there's a number of opportunities in brain-computer interfaces. I can give you an example, of an area where more things are more advanced. So when we or a dog senses an object in the environment, molecules of whatever's being sensed are interacting with protein receptors in the nose, leading to things like opening or blocking of channels. And so and the cell records the current. So we can now design completely from scratch synthetic pores that allow current through, and we can design them so that the amount of current going through is modulated by the presence of compounds in the environment. So we're interested, for example, in making really sensitive fentanyl detectors. So there, the mechanism is really clear. You have a designed protein, it's in a membrane, or we're now making proteins that insert into silicon. And so the proteins in that, which is currents going through, then like a fentanyl molecule or else something else comes along, modulates that current, and you can read that out electronically.
Right. So this is, instead of having sniffer dogs at the airport. Yeah.
Exactly. So we're really interested in that. Could you have something that is detecting fentanyl coming in through packages illicitly into the US? Yeah.
Wow. That is crazy. That's amazing. Early on, you also noted that when DeepMind, announced these breakthroughs in showing how these deep learning systems that throw away physics might be better at doing certain things, you noted how your group and you just sort of shifted. That's a tough shift, right? So there's a certain kind of capacity that clearly you and your group has to be able to, have a flexibility of thought, because many people would say, I don't know anything about those methods. How can I possibly start to do it? So what was that process like?
Yeah, it's interesting when DeepMind announced these results, there was a lot of gloom and doom in the academic community that sort of computational structural biology was just going to be the domain of big tech companies. But I really saw it as very exciting and inspiring. And I think that's part of being a scientist. I mean, that's one of the things that drew me in the first place was the fact that things evolve and change. And the opportunity to really learn something new, I think, was really appealing to me and to my group. And I think people were surprised that how fast we were able to get up to speed on the deep learning methods and then apply them to protein design. And I think that the hard part was actually the protein design thing. You had to know what the problems are. You had to know, how to make proteins. And so DeepMind and other companies had been working on protein design, and it's just a much more open-ended problem. So protein structure prediction is really a closed form problem. Like,
Yeah.
Sequences predict structure, but protein design is really wide open. What do you even design? So, yes, I mean, it's true that we really, it was really great fun to sort of dive into something new, and we were able to get up to speed pretty quickly. But the tools, for developing deep learning methods are quite advanced. Things like PyTorch, the general software is really available. It's main, and you can get a feeling for developing architectures. And one of the things that ML and deep learning are changing so fast. So even if you were a deep learning expert 3 years ago, you're probably a little about a date on the technique. So yeah, there's a certain mindset where you have to be willing to plunge into new things. But I feel like that's a fundamental part of being a scientist. And
But I mean, did you go as far as getting your fingers dirty with writing any code or?
Yeah, so I did. I picked up I just picked up a deep learning project right away, and the code is all pretty high level. If you look at these models have very, very large numbers of parameters, but they don't have huge numbers of lines of code. There's a finite number, and so yeah, that was really fun, and it happened really fast. There was this really interesting transition that took place. We'd been developing these physically based methods, which we called Rosetta, the software, for many years. And there were, graduate students in the group who'd been sort of experts and kind of, black belts in using Rosetta and developing it, who'd been, say, in their fourth year of graduate school. And then there were these deep learning methods coming online. And every couple of weeks, it looked like, they were looking better and better and better. So there was this kind of time of angst where, we were developing these new methods and they looked like they were going to replace the old methods. And then that actually happened. And so there was maybe a few months where people were like, oh, I learned all this stuff and now it's not relevant anymore. But the excitement of being able to make all these incredible new molecules quickly overcame that. And
and then, did you find that?
Yeah.
Did most people in your group acclimate, or were there some?
Oh yeah, everyone acclimated fast. And there are definitely problems still today where the physically based methods are really powerful. For example, if you want to build proteins beyond just the 20 amino acids, you want to incorporate unnatural amino acids. And then for thinking about a lot of these problems in catalysis, you start with physically based models. We do use density functional theory calculations to calculate transition states for chemical reactions that we then build proteins around. So there's definitely a mix of methods being used.
But do you imagine that the deep learning or the deep learning plus that will happen in 3 months or 2 years will ultimately replace those physical methods too? Is it just a matter of time?
Well, it's a little bit hard to say. I mean, so deep learning methods are really powerful when you have large amounts of training data. And I think that's what we've seen with language models and, protein structure prediction. Even weather forecasting is another really interesting case where, there are these quite elaborate physical models, and now the ML models just train on the same data look really powerful. I think that as you extend beyond more and more beyond the sorts of things that are in the training set, physically based models are going to be more and more important. And for the problems that we're working on now, like I said, for the chemistry, physically based models are very important because there's now the DFT calculations are now accelerated by ML. So, there's this gray area,
Right. Yeah. Maybe at some point they'll just be indistinguishable. It'll just be, a blend. But that was one of the questions that I did want to ask. There are some in the large language model skeptic community in the sense that they're very impressed by what they can do, but have noted their view that in a certain number of years, it'll all just move on from that, because they claim that ultimately any of these systems needs a model. It needs an understanding of the world. And so, is that your feeling here too, that ultimately the physics-based considerations will find a home within the most sophisticated versions of these systems? Or will it simply be that physics can be tossed aside and you just don't need it for the kinds of questions that you're interested in?
Again, it sort of depends on the questions that you're trying to answer and how well they're represented in your training set. And I think the physically based things will be important when you're trying to extrapolate way beyond anything you um, have
The data for,
the
yeah,
data for. But
yeah,
coming back to this, the question, the viewpoint of the people that you mentioned, as I said earlier, they're probably all very comfortable with operating with their brains all the time. And writing down how the brain works is something we still have very, very little understanding. Yet we use it all the time, and we don't have as big crises about it. So I think humans, a lot of human endeavor is solving problems and learning more about the world and doing things. And if you have tools that allow you to do more and explore more, they're great. And to some extent, what's under the hood doesn't matter. And I think that's the other answer to why we were able to get up to speed so fast on protein design. We were just really swapping out what was under the hood, and
yeah.
whether it was an ML method or a physically based model that we were using to predict the structure of our design proteins, or to once we had generated a protein backbone, assign a sequence for that backbone, it almost didn't matter. And if we have all kinds of complex hybrid protocols. We're using an AI method here, a physically based method here. And it just, they're almost interoperable parts.
Sure, sure. So just one other quick area that if we could touch on, if you're willing. Obviously we live in a country right now where there's a certain degree of skepticism and reluctance to engage with anything that affects the fundamental workings of a living system, right? Vaccine skeptics and things of that sort. We have a curious state of affairs with the people who are ahead of the health agencies, and various cuts as well that are affecting university research. Have you encountered resistance, protests to the kinds of things that you are working on?
I think that the current situation is very bad, and I think I would say that we've been affected by cuts, but really indirectly. For example, grants that came in through Northwestern, have been cut because Northwestern had grants cut. I would say the biggest danger of the current situation is the effect on the younger generation of science. Current graduate students, even let's say if you want to become a scientist, you want to go to graduate school. In biology, I'm not sure about physics, but in many areas, universities drastically cut graduate school admissions because they weren't sure they'd be able to fund them. NSF fellowships got way cut. So if you even wanted to become a scientist this year, you were a bit out of luck. If you're a graduate student and thinking about moving on to become a professor, all this news about cuts, the opportunities, have been, they're reduced. I had postdoctoral fellows applying for jobs this year who had offers at places like Harvard, which then had to cancel their searches because of cuts. So it's really hard for people at this very important stage of their careers. And so I'm worried about, the effect on really a whole generation of scientists. It's tied into immigration, and so many great scientists and contributors have come from outside the country. And, for the US to really stay a leader in science, it's going to be important that the best and the brightest keep coming to the US. So I think there certainly are problems today, and I'm hoping that the powers that be recognize the importance of maintaining, the both allowing great budding scientists in the U.S. and coming into the U.S. from abroad to move on to great careers. But I think for the U.S. to stay competitive with other countries and just generally to push forward, it's going to be really important to keep things moving and training.
And do you see a trend, either in your group or in the field more generally, of the young scientists saying, you know what, let me just go to Europe. Let me just.
Yes, I absolutely see that. So, whereas up until, fairly recently, the movement was, brilliant people coming into the US. Now people are looking to go outside the US to countries which are supporting science more generously and in a more secure fashion. I think a lot of the problems today are just uncertainty. And uncertainty is very tough on people at sensitive points in their careers.
Yeah, totally. And with the university cuts, and also with, for instance, some of the great work happening in industry, I mean, take Demis as an example and so forth. Is there a pull to industry more than to the academy in the kind of work that you're doing?
Yeah, I think that, I would say that over the years, we've actually started 21 different companies. And that's because that comes about because students, when they develop a new protein, something that could cure cancer, they get attached to it. And we have a translational program so they can attract, develop to the point where they can attract funding, start a company. And so there's been a lot of interest in starting companies, and that has actually grown in the last, recently, with people becoming less optimistic about careers in academia. And I think, that's great, but obviously it's important that we have a next generation of scientists in academia as well.
Yeah, well, let's hope that message resonates broadly, because
Yes.
we're all under that same kind of pressure right now. Well, David, thanks so much. This has been a fascinating tour of the groundbreaking work that you and your colleagues have been doing. I love talking to people like you, because at the end of the conversation, I say, wow, I'd love to do that. It's so exciting, to hear about the kinds of work that you guys are doing. So thanks for spending the time with us today.
Yeah, thanks, Brian. That was really fun. Yeah.
Excellent. Bye.
Okay. Bye.